Hello, everyone, and welcome to Biohackers Lab. I'm your host, Gary Cohen, and on today's episode, I have Dr. Michael Lustgarten PhD. Dr. Lustgarten is Scientist 2 in the Nutrition, Exercise Physiology, and Sarcopenia Laboratory at the Human Nutrition Research Center on Aging at Tufts University. His research currently focuses on the role of the gut microbiome and serum metabolome on muscle mass and function in older adults. He has contributed to 22 published articles in leading peer-reviewed journals that have been cited thousands of times. He is also the author of a book called Microbial Burden, a Major Cause of Aging and Age-Related Disease. Michael, thanks so much for coming on for an episode for today.
Thanks, Gary. Thanks for having me on. Looking forward to it.
Yeah, so am I, because I just read your book that is on Amazon on Kindle. And then, of course, I've actually been trying to follow you a little bit on Twitter and just seen the things that you're sharing on your Twitter profile there too. And I've had a previous guest, Amy Perel, which we both know her, and we talked about the microbiome and how viruses and funguses and bacteria influence our health. And Jay, you wrote and you're studying how these things also influence our aging, and you're then doing some very interesting self-personalization testing too, which we're going to get into the episode for today. So if you wouldn't mind just introducing listeners, how does the microbiome affect the four leading theories of aging, and what are those theories?
So when you say, well, it's actually four of. There's something known as the hallmarks of aging. So what I went over in the book were just a few of them, but them being telomere shortening, oxidative stress, inflammation, and what else? What else did I put on that list? Mitochondrial dysfunction, maybe.
Yeah, and insulin resistance.
Insulin resistance, right? So there's actually a lot more than that, like protein homeostasis, DNA damage, and actually in the next iteration of the book, I intend on showing all the data that shows a role for microbial burden as underlying all of the hallmarks of aging. So, so I guess to get into that, how microbial burden affects those things, we should take a step back into what happens in the gut, just as an example, because these changes during age or the passage of time, because actually the definition of age, I think, can be blurred once you start thinking about it in this microbial context, but beyond that. So in the gut, the gut is basically a tube, and it's lined by epithelial cells. So the integrity of the epithelial cells to not allow stuff to enter them and enter the bloodstream gets worse with age. So gut barrier function gets worse with age. As a result, stuff that's inside the intestinal tube gets into the blood, and then some of these things are microbial products, some are dietary antigens, things that can cause inflammation. But nonetheless, some microbial products, as I talk a lot in the book about, one of them being lipopolysaccharide, which is found in the outer shell of one specific type of bacteria, gram-negative bacteria. That's how they stain. So lipopolysaccharide is pretty well studied, even though, as I'm sure Amy may have, or I don't know, maybe she didn't, but there's gram-negative bacteria, there's gram-positive bacteria, and then you've got fungi, viruses, all kinds of microbial stuff that can activate our immune system. But lipopolysaccharide is just one that's been thoroughly studied. So when lipopolysaccharide is increased in the blood, that actually causes an insulin resistance. It actually stimulates the mitochondria to increase superoxide production, to increase oxidative stress. And these are two of the leading causes of aging. LPS binds to receptors on cells that trigger inflammation. So we've got three major, I think with it, as I discussed in the book, I think with an increase in viral load, there's an acceleration of telomere shortening. So that's the quick and dirty on microbial burden affecting just those four mechanisms of aging. Now, what's interesting is that high caloric diets, and I'm not going to say high fat diets, because even though that's how it's been used, I don't want to talk negatively about the macronutrient composition, because I think it's more of a focus on high fat diets lead to overeating, and at least in rodents, and an increase in caloric intake, which is part of the problem here. So a high caloric diet, at least in rodents, stimulates this breakdown of the gut barrier, thereby leading to an increase in the microbial products in the blood, triggering these things that negatively affect us and accelerate aging. So so that indicates a role for diet in this process, independent of aging. So basically, you can accelerate aging, which is basically what obesity is. It's an accelerated aging phenotype. So, yeah, that's the quick and dirty of this.
Yeah, no, I'll give you a loaded question. That was a big one to begin with.
Sure, yeah.
But I think already what I'm hearing here is how the health of our gut lining can help determine how well we age.
Yep, definitely. And as I write in the book, one major strategy would be a very high fiber diet, because our intestinal bacteria, 99% of our gut microbiome is bacteria. So you want to feed them what they like. And at least evolutionarily, there's data showing that our diet contained about 3.3 grams of fiber per 100 calories. So if you do the math on a 2,000 calorie diet, that's 66 grams of fiber, whereas the RDA is it's recommended 20 to 30 grams. So it's paltry compared to how we evolved. So even if you just go by the evolutionary dietary fiber density, that should our intestinal bacteria will degrade the fiber into short chain fatty acids, one of which is butyrate. Butyrate is actually involved in mechanisms that improve gut barrier function. So I propose that as a major strategy in the book for improving health and potentially lifespan.
And that also then leads into one section where you were talking about protein intake, weren't you, in the book, and saying that higher protein intake may be associated with lower gut barrier function?
Yeah, so that's a little more complicated now. And since I've written the book, I've reconsidered that. I still think that very high protein intakes, I'm not talking about 100 to 150 grams, but maybe 200. I don't have an exact number, but the fact is, in rodents, protein restriction extends lifespan. And one mechanism that probably does that, as I note in the book, is that is by improving barrier function. So, but there's a flip side on that. So, protein, if you don't have enough protein, you're not going to have enough muscle mass. So muscle mass is possibly associated with lifespan. And then there are specific amino acids that actually improve gut barrier function, one of them being glutamine. So this goes to back to the effect of the quantified self, and how would you know if your protein intake is too high or too low? And what's interesting is that even with normal kidney function, how much urea you've got circulating in your blood, it's a metabolite known as blood urea nitrogen on a standard blood test, is linearly proportional to how much protein you eat, assuming normal kidney function. If you have kidney disease, all bets are off, because your urea, circulating levels of urea will be higher than normal. But the reason why this is important, because if you're eating a ton of protein and you're making more urea, and if you're not peeing it out, urea actually degrades gut barrier function. High levels of urea, well, actually, it does two things. One, it stimulates the growth of urea degrading bacteria. And actually, that enzyme, in the presence of urea in the intestine, degrades intestinal barrier function. So I can see how a very high protein diet can negatively affect lifespan through the gut. But alternatively, a very low protein diet, we're not, I'm a scientist, I'm pro rodent studies. These are important models for studying aging, but these, methionine restriction or protein restriction diets are done on mice in a cage who are sedentary for their entire lifespan. It's basically a jailhouse diet. You're stuck in a cage forever. You're given a standard diet, can't exercise, you can't move. So in that case, obviously it may have some benefits, but this is not the majority of us in the population. So, as I mentioned, too low of a protein diet too, whereas it may extend lifespan in these caged animals, it can negatively affect the immune system. You're not going to produce enough lymphocytes, which obviously are important in immune defense. So finding that window of not too low, not too high, which goes back into the effect of using blood testing, which I do, to make sure that your blood urea nitrogens are not higher than where they should be, within a nice little range, not the reference range, but what's actually been shown to be optimal in terms of mortality risk. And the reference range is not always indicative of that.
So could you just explain to people then, because I'm going to ask a couple other questions related to the fiber and the protein, because I know I've got a lot of listeners who like to follow a carnivore kind of diet, and that's, a heavy meat-based diet. And
yep.
And I know they're going to be going, Gary, please ask Michael this, please ask Michael that. But I also want to tie this into what you just brought up there, where you're using blood tests to try to determine your ideal point. So
Yep.
could you just explain then, if someone wanted to do blood tests themselves, or if they just listen to how you're doing it and how you're determining using that?
Sure. So for protein intake, it's going to sound very reductionist, but right from the start, I want to preface it by saying, whether you're on a carnivore diet or vegetarian, vegan, omnivore, whatever it may be, or flexitarian, I advocate looking at the broad range of blood test markers. So if you're just looking at your ketones, triglycerides, insulin, glucose, lipid profile, you're shortchanging the kidney function, liver function, immune function. I'm looking for the whole context. So if my protein intake is X and my blood urea nitrogen, knowing that urea production is linearly correlated with protein intake, if I'm only looking at my blood urea nitrogen, but all my other biomarkers are out of whack, well, I've got a problem, right? So when I say that I use my protein intake and I look at my blood urea nitrogen, that's also evaluated in the context of all the other biomarkers being where I want them to be. So I don't want anybody listening to think, all right, I looked at my blood urea nitrogen, it's good, but all right, what does your other stuff say? Because if your other stuff is out of whack, then you should probably add some stuff into your diet or take some stuff out. So blood urea nitrogen is a simple blood test. It's found on the standard blood chemistry screen, which is a $35 test. I don't go to my GP to get it done because if I'm there 4, 5, or 6 times a year, they're going to say, I'm not doing this test for you. Insurance is going to cover it. And then I'll get charged $200, $300, $400 out of pocket, which is ridiculous. So I go online, they send me the blood test, and then I send my blood back to them and they send me the results in 2 days. So for blood urea nitrogen, the reference range is 5 to 20 milligrams per deciliter. But then again, it's not about the reference range. It's what's been shown to be optimal in terms of risk of death from all causes. And actually, how does it change during age? So blood urea nitrogen goes up during age. So that's one thing to track. I mean, if your protein intake is at one level and you see your urea levels going up over time, well, your kidney function is getting worse. But assuming, again, going back to say that assuming your kidney function is normal, and if you're increasing your protein intake to whatever value, you want your protein, the data suggests that blood urea nitrogen between 5 to 15 is optimal based on the all-cause mortality data. So higher than 15, there's actually a higher risk of death from all causes. So that's where I try to keep it. Actually, my blood urea nitrogen is about 10 to 11, and I had it once upon a time. I went vegan, and that wasn't optimal for some of my blood test stuff. So I increased my protein intake. And actually, what's interesting is homocysteine, which is a marker of cardiovascular health. It's also related to stroke and dementia risk. My homocysteine levels are higher than they should be. Optimal is between 5 to 9. Mine were at like 11, 12, and as high as 15. So, I tried the stuff that's published in the literature without going into too much detail. And then because I track my diet every day, I weigh all my food. And then because I've got like 15 to 20 blood tests over a 3-year span, I can look at my average dietary intake with my blood test variables and the correlations between my diet with the blood test stuff. And what I found was my protein intake was actually most strongly correlated with my homocysteine levels. So, within that 5 to 15 range for the blood urea nitrogen as a marker of protein intake, I was at 5. So I said, well, let me increase it because I've got some window there. Let me increase it. So I did. And actually my homocysteine went down. So that correlation may have been a causative thing, whatever the mechanism. So right now my blood urea nitrogen is between 10 and 11, 10 and 13. So I'm still within that 5 to 15 milligrams per deciliter range. But I also keep an eye on my kidney function and all the other stuff too, to make sure that, I don't fix one thing and mess up 10 other things.
And then also how I introduced you, that you're working with sarcopenia, and I've had Dr. Stuart Phillips on, and he's a big advocate that we actually need more protein as we age too, to be able to avoid things like sarcopenia. So, and as you said, as we age, typically our kidney function degrades naturally anyway. So we're in this situation where we need more protein, but our kidney function is also going down. So how are you handling that, knowing what you know about sarcopenia? Are you making sure that you get enough protein to avoid sarcopenia, but then also improve your aging?
Yeah, definitely. So what's interesting is I just submitted a grant 2 days ago that looks at the kidney gut muscle axis, and that that story involves decrease in kidney function, alterations in the gut microbiome, and increased fat and muscle. So with age, there's more fat and muscle. Your muscle composition gets worse. So, yeah, I'm keeping an eye on my kidney function stuff, which is all normal or close to where it should be for biologic youth. But what's interesting about that protein story too is going back to the idea of too much. So with a very high protein intake, and again, that's going to be variable. For me, I don't know, maybe 200, 250 grams. For someone else, maybe it's 150 grams. It's variable. The more protein you eat, more of it will slip from not getting absorbed in the small intestine and get into the large intestine, where it can then be fermented by gut bacteria. And the gut bacteria, colonic bacteria, the composition of your colonic bacteria will depend on your environment, meaning diet, exercise, air quality, everything. Whether you have a viral infection in your nose, that can affect it too. So if your protein intake is very high, you can increase the composition of protein degrading bacteria, which can produce some of these things that actually worsen kidney function. So for those who are interested in looking at how their protein intake is affecting their physiology, I think it's important to look at your gut microbiome. Fortunately, I haven't seen any of those, an increase in protein degrading bacteria in my intestines. So we're good there. But those protein degrading bacteria also increase with age. Probably one reason that older adults may need more protein, right? Because if you're eating more protein and you're 75 years old and less of it's getting absorbed in the small intestine, some of it's getting into the large intestine, and now you've got more of these E. coli or other protein degraders. That's not good. So yeah.
Okay. Yeah, because I'll come back to some of those questions then that I know people want to be asking me. And I'm just thinking here, if we probably stay with the protein one and talking about the microbiome of your guts and gut lining, I've had guests on the show who appear to have very bad intestinal permeability issues, which is causing autoimmune responses and a whole cascade of conditions. And when they adopt mainly a meat-based diet, then it seems to solve a lot of those problems. So in this case, and I've had another lady who they run a clinic in Hungary called Paleomedicina, and they focus a lot on just eating fat and protein to try and improve intestinal permeability. So it's interesting to hear that on one hand, we're looking in the research that says that protein can cause intestinal permeability issues, but then we're hearing case studies of people adopting just more fat and meat to improve intestinal permeability.
So I don't want to say that the autoimmune, for example, Jordan Peterson's daughter, I know that and actually Jordan Peterson, right? They went all meat.
Mm-hmm.
But I'm not trying to say that my way is how everyone should do it. I do my own testing. I'm trying to find what's optimal for me. But maybe as a TMI and too much information, I grew up on a terrible diet, eating like boxes of cakes and boxes of cookies, and that's what I would eat in that day. And I don't want to blame my parents, but they're the ones who bought it. So, but then eventually in my early 20s, I adopted that way, and I was like, what do I need to eat? Vegetables or fiber? I'll eat just crushed protein bars, whatever. So that led to hemorrhoids and bloody hemorrhoids. So, and that's intestinal inflammation. So maybe it's TMI, and if people don't like to hear about hemorrhoids, they can tune off now for, come back in a couple minutes. But I can say that I've gone vegan. I've gone on a super high fiber diet, like 130 grams a day. I still average about 100 grams a day. But I noticed that, including about 300 calories from chickpeas, which all the long-lived societies eat beans. So, it's one thing to use the epi-data, but what does the epi-data say in your own physiology? So since I've flipped that out for, 5 to 7 ounces of grass-fed beef, thereby lowering my fiber intake for the day, still, I'm at like 80 to 100 grams. I'm still way above the RDA. That issue with hemorrhoids has dramatically subsided. So, and usually the therapy for once you've got a hemorrhoid is you've got to band it off. I'm not trying to get, have scars in my intestines. So I can see a role. It's an individual based thing. I'm not a fan of people who go vegan and say, oh, I feel great. Well, what do your blood markers say? And I'm not alternatively someone who goes carnivore and says, oh, I feel great. What are your blood test values? What does the data actually say? And I've seen some of Michaela Peterson's blood test stuff, which looked pretty good. I mean, I think she had a couple things that were out of whack, but these are easily fixable things. I didn't see anything blatantly obvious, which you would expect from eliminating all the other food groups. But yeah, they may have some genetic issue that that's optimal for them. So
yeah, and again, I know I'm just asking these questions because listeners are going to want me to ask this stuff, but I'm also very pro N equals 1. Like as you were saying, use your own body, use your own physiology, subjective or objective, and just say this is what makes me feel best, and that's what you're finding in your case there. So,
but also the data to back it up, and not just like, oh, these are my ketones or triglycerides or triglyceride to HDL ratio, which I actually see a lot of pro, meat, eat only meat, carnivore only diets. I see them focusing on, just a few biomarkers of cardiovascular burden or cardiovascular health, and it's the full picture. It's full of the picture as you can get, because it's one thing to feel good, but then it's another thing to say, all right, here are my biomarkers, and that's why this works best for me. I bet that pure carnivore isn't going to be optimal for most people. Including a little bit of vegetables that they can tolerate will be optimal, whether it's gut barrier function or other stuff. But yeah, most people don't measure their systemic inflammation, which is important. So
we touched on it a little bit earlier as one of the theories of aging was insulin resistance. So I'd be interested with your own personal quantified self-tracking. Are you tracking insulin resistance or your glucose, your ketones, or something in that respect too, to make sure that you're in a more optimal place?
Yeah, 100%. So I haven't tracked insulin levels, but I have, like I said, glucose levels, going back for 20 years. And as I mentioned too, like I besides one blood test a year, which at best most people would do, I really wanted to get a systemic understanding of where my body was. So I did it like 15 times within a 3-year span. So yeah, my blood glucose levels are pretty consistent. I think my average is 85 over those 15 or so blood test measurements. So, but then that being said, you could have normal blood glucose and your insulin levels have been going up during time. So I'm considering, including insulin into my list. But yeah, I'd say insulin resistance is probably one of the major markers of poor aging. If you're insulin resistant, you're on a bad track and you get off that quickly. Especially since you can tie it back to microbial burden. So yeah, I'm on top of that. My glucose levels, good.
Okay.
But then again, it's not just related to my diet though. I tried, I walk 10 to 15 plus miles a week. I strength train 3 days a week. I'm fit and active. So that may play a part in it too. It may not just be diet. So.
And on your blog, on your website, you also do you've done some interesting experimentation where you've tried to adjust your gut microbiome to improve your deep sleep, I think it was. Yeah. Could you just tell people about that experiment?
Yeah, so there is some data on the gut-brain axis, and it's evolving. And I'd be shocked if there wasn't a strong role for the gut on influencing brain, whether it's sleep or anxiety, depression, whatever it may be. And bifidobacteria are they produce short chain fatty acids, which can be involved in gut barrier defense. And I postulated an important role for them in aging. They decline with age. They're very high in infants, and they decline all the way down to almost zero. So I've had almost zero bifidobacteria in each of my 4 microbiome measurements. And one reason maybe is the antibiotics I took as a kid. I mean, my parents had antibiotics, when I got sick or whatever, and had them on the shelf. And, my dumbass at however old I was like, oh, I don't feel great today. So I popped some antibiotics, which is mental. It's so ridiculous. I can't believe I did that in hindsight. But I have no bifidobacteria, or had. So at least on Ubiome, before they got raided by the government, they had on their website that bifidobacteria may be involved in sleep quality. And one measure of sleep quality is the amount of deep sleep you get. When you wake up in the morning, if you feel terrible, you may have slept 8 hours, but got almost no deep sleep, which is the slow wave sleep, delta sleep. So, so considering I didn't have any bifidobacteria, and there was some published data linking metabolites produced by bifidobacteria to sleep quality, and knowing that like 10 years ago, I did a polysomnograph, which is a sleep study, an official sleep study. You go to a sleep doctor and they measure your sleep stages. My deep sleep was like 5% of total sleep, which again is like an 80-year-old. When you're young, you get about 20%. So yeah, I started tracking my deep sleep with a fitness tracker. And granted, fitness trackers aren't going to be as good as the gold standard of tracking your sleep in a sleep study. But the error should be systematic, meaning whatever the error is, it should be across the board. And I'm going to get relative amounts. So yeah, I tracked my diet. I tracked when I took the probiotic, which in this case was a bifidobacteria mix, and I've tried to get the bifidobacteria into my gut 3 different times. So it's a dose thing too, that I get the right probiotic, that can survive the acidity of my stomach. So tracking diet, taking the probiotic, and then tracking sleep, and then looking for correlations there, which is what I published on my blog. And actually, I saw after I stopped taking the probiotic, I saw a spike that I didn't see for 4 or 5 months of tracking my deep sleep. Now, whether that's related to the bifidobacteria, I don't know, but my bifidobacteria went up a bunch. Even a couple weeks after stopping the probiotic supplementation, which suggests that there may have been a role for the bifidobacteria on affecting my deep sleep. But that's still a work in progress. I'm still obsessed with getting as much deep sleep as possible. Unfortunately, my fitness tracker, they changed their algorithm. So I had 240 days of tracked sleep data with my diet. And now it seems like I'm getting double the amount of deep sleep I was before, but I'm getting to sleep earlier. And so now I have to get another 200 days of data to see, so it's still a work in progress. Whether or not the alterations in my gut microbiome did it, it's unknown, but it's still an interesting find that, the increase in my bifidobacteria went up correspondingly with my increase in deep sleep, at least during that time.
Mm-hmm. So, yeah, now I'm bringing up sleep too and microbial because I've had Dr. Ben Bickman on, and he's talked about insulin resistance, and he was mentioning how like if you don't sleep well, also that can contribute to insulin resistance. And so this is all tying in that how we mentioned if you have insulin resistance, it's with the microbial, and it's an age-related thing, and so we need to try and get optimal sleep as we age too. But my question always when I think of this, and I know others have asked, is so. When you see a probiotic on the shelf in a health store, like how much of that can I actually use in my body, as you said, because a lot of it gets destroyed when it first hits your stomach lining. So even in this case, are you a fan of certain ones like live or, the ones you find in the fridge, liquids, powders, any sort of guidance on anyone if they wanted to try and experiment themselves?
Yeah. Step 1 is get your gut microbiome analyzed. That's got to be step 1, because if you're just going to take a probiotic cocktail, whether it's lactobacillus or bifidobacteria, whatever it may be, without knowing what your actual composition of your gut microbiome is, because if something's got to live in there, something's got to get out. So maybe you take a probiotic and push out a better symbiont. So first step is to get your gut microbiome sequence. Second step is to figure out if there are things that you want to increase with some hypothesis, right? If your gut microbiome looks like what youth should like and doesn't look like an 80-year-old microbiome, but then granted, it's complicated, right? Because most people maybe don't look that stuff up, but I'd encourage everyone to look into these things, because I'd say blind supplementation is just may do more harm than good. So step 1, get the microbiome sequence. Step 2, figure out if there are bacterial species, assuming it's bacteria that you would want to change there, that you would want to increase. Step 3, actually get your microbiome sequenced again to see if they're there. Most people don't do any of these things. They take it, they say, oh, I feel great, but it could be a sugar pill placebo. You don't know until you've actually got the data. And then step 4, track some outcome measure that shows that taking the probiotic is actually doing anything. But you're exactly right in terms of whether it's in liquid form or the dose of it may be too low to get in there. It's a trial and error process. Now, unfortunately, I'd say most people don't try enough to positively alter it with probiotics. I'd actually put probiotics down lowest on the list, unless you had some extreme situation. I'd say changing your diet probably is step 1, assuming that most people have a normal composition that you can change the levels through diet with. But in my case, I had almost zero bifidobacteria. So I could have, so prebiotics are defined as foods or food items that feed certain species or strains of bacteria. They're enabling them to flourish. But if you don't have those species or strains in your gut, they can't grow. I mean, it doesn't, so you need the seed. So I had the seed, which was the bifidobacteria. And then after, X amount of days, whether it was 3 weeks or 30 days or whatever it was, I was like, I'm going to stop taking the seed. The seed should be there. I'm just going to give them the prebiotic that they like. So probiotics shouldn't be indefinitely. If you're taking them indefinitely and they're not seeding your gut, you're not giving them the prebiotic that they need. So there's a lot of factors there that people should really dig into if they want to seed their gut properly. But first step would be foods, I'd say, finding out which foods. Actually, first step would be finding out what your composition is and how you want to positively or negatively change that.
Mm-hmm. Yeah, no, and when you talk, sorry,
real quick. Even especially if you're on a meat-heavy diet, you want to see if you've got an increase in the protein-degrading bacteria. And for an example, it's very simple and straightforward. It's proteobacteria, but not all proteobacteria are created equal. Within the proteobacteria are a family called enterobacteria. These are the urea degraders. They're bad guys, quote unquote. If you've got too many, if your meat intake is too high and you've got a ton of enterobacteria, you may want to consider cutting down on the meat.
Okay. And yeah, when you're talking there about the seeding element too, I think of seeding either through your mouth, and of course there's now the new age of fecal transplants as another source of feeding. And I guess people are then considering what is the more, if you do want to alter your ratios, what's going to actually be able to do that. And it seems like, I don't know, maybe fecal transplants are going to be more powerful than taking a probiotic if you're at that stage. Is even someone like yourself who's into quantified tracking, have you ever even considered something as crazy as trying to go for a fecal transplant to increase a ratio?
Oh, yeah. I've considered it, but I haven't done it. But what's interesting is there's a scientist, Laura Peterson. She published a paper on the microbiome in cyclists. And if you Google her name, her story should come up where she actually got a fecal transplant. I guess she had some massive GI issues, and I don't remember if she took antibiotics or however she did it. But yeah, she talks about how she did an FMT, fecal microbiota transplant, and she says that it improved her health, like her gut issues went away. But the issue that I have with that, I mean, maybe in some situations it would be beneficial. Like as it's used now, it's used for people who have Clostridium difficile infections, which is a nasty infection. It could cause death. So it's a life-saving transplant. But you see, the composition of your microbiome is going to be dependent on what metabolites your cells are making in your intestine. So on the one hand, if I can see in the short term where, let's say that you wipe out your microbiome temporarily, completely with antibiotics, just wipe it out and then you do the transplant. So someone else's microbiome should colonize your GI tract for some small amount of time in the exact composition that came from the donor. But then that microbiome is going to adapt over time based on your genetics and based on the metabolites that you produce, and based on your environment that you're exposing it to, whether it's diet, whether it's exercise, air quality, as I mentioned, all those factors. So without actually trying to address some of the underlying root of why you would want to get a fecal transplant, in the case of people who have C. difficile infections, it's a life or death kind of thing. But if you're just trying to do it, so if they don't get it, they can die, right? But if it's for someone who just is interested in optimizing their health, they may want to address the underlying issues first. Because just as a quick aside, what I didn't mention is that I've got a paper in review where we transplanted fecal samples from older adult humans into germ-free mice. And that model has established a causative role for the microbiome on things like obesity, on kwashiorkor, immunosenescence, a whole bunch of outcomes. So, but these have been done in young animals, basically transplanted from young to young. So we did it from old humans into young germ-free mice, and we actually saw a difference in grip strength in the mice, which would suggest a role for the microbiome on muscle strength as a causative role. But some of the microbiome didn't transplant and may have changed over time. And the reason I bring that up is because the only other study I know of that did an old transplant into young it was from mouse to mouse. They saw that 1 week after the transplant, it looked like an old donor in the young animal. But after a month of having the old stool, it was just like a young microbiome. So an animal that didn't have a microbiome got a stool sample transplant from an old animal. 1 week it looked the same as an old animal. After a month, it adapted to where it was like from young animals. So I predict that young people or young healthy people getting these transplants, that's probably what would happen. It would just adapt over time. So if you don't change your diet or your lifestyle, you haven't addressed the underlying issue, and your biome would change back to where it was. That's what I'd predict.
Yeah, I'm just thinking, I don't know if this is even talked about in the longevity anti-aging conferences where you've got people trying to do fecal transplants for longevity reasons. And this would be I guess that's a valid point. If you're younger or below a certain age and don't exhibit certain things, then you probably will gain no benefit from doing that. If you, that level of trying to stay young, it's maybe only beneficial for certain people at a certain age.
Yeah, in a certain condition, yeah. So unfortunately, we live in a big pharma-based world, in a drug mentality for improving health and longevity, whether it's senolytics or other stuff. And what we do know, though, is a good diet, staying lean, and exercise, staying fit, probably will give you 15 to 20 years of increased lifespan above the average lifespan. Whether to get you to the maximum lifespan, 122, that you don't see marathon runners or bodybuilders getting that far. But to get from 75 or 80 for a man and a woman, respectively, to get to 90, 95, based on at least the rodent data and based on data in Olympic athletes who live longer, it's pretty known. So I'll take that trade-off versus taking a pill or a fecal transplant or, this reductionist view. Will you get 6 months more life? I don't know. Will it shorten my life by 6 months? So, most people don't want to go through the diet and exercise path to get 15 to 20 years. I'd put that first.
Mm-hmm.
So, but eventually, because I have no interest in death, I can't stand that idea, or functional decline. We're going to need some cellular rejuvenation to fix our old, aging cells. So, proper diet and lifestyle can only get you so far. But using the drug-based approach or probiotic-based approach, that can only get so far too, with severe limitations, I'd say.
Mm-hmm. And what you were talking about, where you transplanted from an old donor to a young donor, and then eventually the young also overpowers the old donor's microbes.
In my study, we didn't. Sorry to cut real quick. In my study, we don't know if that happened because we only measured at one time point. But in the old into young mouse into mouse, they saw that. Yeah, they saw that over a 1 month period with 2 time points. Yeah, they saw that. Sorry.
Yeah, no, but because what I also think about is, because a lot of the time when still I'm in the habit of doing that, when I think of the microbiome, I just think of bacteria, but it's not just bacteria. It's viruses and funguses, and there was another word that Amy used in our interview.
Archaea.
Yeah. and so, but at one point that she did bring up too was that how viruses infiltrate the bacteria too in our system. So in that case, if you take an antibiotic, you may get rid of the bacteria, but you don't deal with the viruses and you don't deal with the fungus. So the host still has those elements to their microbiome, even though you're trying to only influence the bacteria side of the microbiome.
Which would go into the idea of the transplant whole biome, you would get around those effects. But then, as I mentioned, if your genotype is whatever your genotype is for having a certain microbiome, now you've temporarily changed the biome with someone else's biome, but your genotype to have a certain biome is still there. Your genes are going to make certain proteins and your cells are going to make certain metabolites, and that's going to be the fuel to grow a certain metabolism. So temporarily you may change that with someone else's microbiome, but over time I predict it will go back to the same, as it was before, unless you change the input variables, whether it's diet or exercise, things that are known to positively affect the microbiome. But to that point too, that it isn't just, we have these individual species of different microbes, whether bacteria, fungi, virus, archaea, whatever. They interact with each other and coexist with each other. So it's basically like a neural net. This bacteria is going here. The metabolites they're making are the growth for a lot of other microbes. So right now we're just in the infancy. First, it started with whole scale sequencing of the bacteria that are down there, and then it's evolved into what are the viruses that are there and what are the fungi that are there. But these interrelationships of the entire microbiome, whether it's in the gut or skin, we're still unfortunately in the infancy of it. And eventually, I think it's been estimated that there are enough microbes down there to be similar in size to the human brain. So I'm sure that there are neural nets, how they interact in terms of how they interact. So we're going to need that kind of computing power to learn about the interactions, learn about how a particular thing, whether it's a probiotic or diet or exercise, affects the entire community, the entire ecosystem. And then after evaluating that intervention, does that positively or negatively affect the complete ecosystem of your microbiome? That's where I see this going. How fast do we get there? That's another story. But right now, we're still just barbarically doing it. Take these probiotics and see what happens, right? But eventually, the tools will be more specific, whether that's 10, 50, or 100 years, eventually. So.
Mm-hmm. And a point I want to also touch on that you brought up in your book was about our adipose tissue. So how the amount of fat that we have as we age and how that actually has a protective mechanism for us as we age. Could you just educate people about that?
Yeah, I think that's very interesting. A very interesting finding that's dramatically understudied. And actually, I pitched the grant to the NIA, National Institute on Aging, last year, that I just didn't have enough preliminary data. But it was based around this idea that fat cells produce antimicrobial peptides. And that evidence comes from skin, fat cells located in the skin, under the skin, subcutaneous fat cells that when exposed to a gram-positive bacterium, in this case, Staphylococcus aureus. So, when you see an athlete who has a hole literally in their leg, flesh-eating bacteria, that's Staph aureus. They're nasty. So when you expose fat cells located underneath the skin, subcutaneous fat cells, to Staph aureus, they actually make antimicrobial peptides, one of them being cathelicidin. So in the grant that I pitched last year, I proposed that when considering that microbial burden is increased in older adults and that we have an increase in fat cells in our muscle with age, I pitched the idea that the increase in fat cells in muscle with age may be serving an antimicrobial role. Now, we still don't know. I'm still going to go after that in future grants. But so then it goes back to this idea that obese people actually have higher circulating microbial burden. This has been shown in rodent studies. It raises the issue, is that a compensatory mechanism to improve immune defense against microbial burden? And I'd argue that probably yes, especially when you consider that a high-calorie diet weakens the gut barrier. There's an increase in circulating microbial products, which is probably overwhelming our immune cells, which circulate. And then one response that our body can use is by increasing the size and amount of fat cells. And considering the fat cells can make these antimicrobial peptides, they're probably helping with our immune system. So there's that idea is just emerging. There's only a few studies who have actually studied this. But yeah, I pitched that idea in the book, and I'd say it's based on solid footing in the rodent study. So I'd be shocked if it doesn't turn out to be the truth when it comes to human data.
So I'm interested in yourself again with your mission to, optimize your longevity and your aging. How are you handling that aspect of it, knowing that potentially fat is a good thing to have as we age too? Or is it that we have to stay within a certain body fat percentage, potentially that, or if we see that it goes up too high, actually that's an indicator that we've got microbial burden and that we need to deal with that to improve our adipose tissue or body fat percentage?
Yeah, so that's a double-edged sword, just as you mentioned. So I'd say this first step is staying lean and making sure that your markers of inflammation are low for the duration of your lifespan. And if they start to change, whether it's like you said, if your amount of fat goes up and you're not as lean as you were and your inflammation is going up, you probably have an increase in microbial burden. What that microbe is, whether it's bacteria, fungi, virus, whatever, you've got to figure it out through independent testing. But first step is staying lean. And I'd say that the increase in fat during aging, again, is the compensatory mechanism, whether it's your caloric intake is too much or you're not giving your gut the proper nutrients that you need. But there is actually data in older adults showing that a higher BMI is associated with a lower risk of death from all causes. But in that cohort, they probably weren't very lean or fit to start out with. So it's like on a standard American diet, which isn't very good, having more fat will probably give you a longer lifespan than someone who doesn't. But the flip side of that too is muscle isn't the only thing that goes away during aging. Fat does too. And, I don't know, maybe it's just my bias, but you could make the argument that when you consider that the role of inflammation has on negatively affecting muscle mass, and inflammation can cause decreases in protein synthesis, leading to decreases in muscle mass. it may be that these microbes can also, turn off stuff in our fat cells to stop making antimicrobial peptides. So now we're just on the rapidly accelerating slope to death because the muscle's gone and the fat's gone. It's end of life. So as I said, I mean, it seems like I just packed a lot of information in there. First step is staying lean. If you see that your body fat is starting to go up, and your inflammation is starting to go up, you probably should address what the underlying issue is there. And if it's a high-calorie diet, that feeds back into this idea of high-calorie diets worsen gut barrier function, probably leading to an increase in microbial burden. But and if you're not tracking your calorie diet and don't do anything to fix these things, I mean, I'd say that's a blind approach to health. So it's important to track these things. But yeah, hopefully I unpacked some of the story there.
Yeah, no, you did, and I know it's a complicated subject when we start going into microbiome and the links to all the systems, but it's a fascinating topic because we're meant to be in a symbiotic, in a beneficial relationship between our body and these other little guys that are traveling with us as we go through life, and we're just trying to figure out now how do we optimize that relationship across all our different organs.
So, sure. Here's the drawback, though. Germ-free mice, animals that don't have a microbiome, bubble mice, they're grown in a bubble for the duration of lifespan. They're not immortal. So there still is cellular aging independent of the microbiome. So you could say, well, then the who cares about the microbiome? But the fact is, that shows that the microbiome can affect how long you live. But if you don't fix, so considering that our cells age and make different metabolites that go on during aging, our microbiome is going to change in relation to that composition. And even if you're doing all the right things to keep yourself healthy and fit and live long, considering that cellular aging will change independent of the microbiome, we've got to go after things that actually address that issue first. But yeah, you can slow it down. You can slow down. You can keep your cellular function closer to a youthful state, which will keep your microbiome closer to a youthful state, potentially giving you more and healthier years of life. Yeah,
and the key thing here is I've talked about it on the show with others before, is about mitochondrial health, so our microbiome can influence our mitochondrial health.
100%, and this is most people don't know much about this, and I think this is so. I did my graduate work on mitochondria, so I started off thinking they were the root of aging, life, and death, and they essentially are. You don't make energy, you die, right? Mitochondria consistently making energy, for example, in the heart. Mitochondria don't work, your heart won't pump. So what's interesting about that is, as we mentioned in the beginning, that mitochondria produce superoxide, and which is toxic. It's intended to kill stuff. So that's one way that our body can address microbial burden. But what's also interesting is that circulating mitochondrial DNA increases during aging. So, on the one hand, you could say, mitochondria function and mitochondrial health gets worse with age. So their DNA is in the cytoplasm and it's in the blood. What does that have to do with anything? So there's actually studies showing that the mitochondria eject some of their DNA as a signal to the immune system to target the microbes. So I just think that's amazing, an interesting finding. So that's also on my list, is to look at, circulating mitochondrial DNA as a marker of microbial burden.
Wow.
Yeah.
Yeah, I can imagine that test then. That's what I start thinking of, we start testing that level, to see okay, so it's showing our mitochondria is part of our innate immunity, is actually trying to then send a signal to the body, we need to deal with something here.
Yeah. And I bet they release it's not all of their DNA, they release parts of it, but I bet they release certain parts of it in response to certain microbes. And especially when you consider the mitochondria are located in the cells. So microbes and their products don't just circulate in the blood. I mean, granted, macrophages are one of our immune cells will engulf and phagocytose these microbes to kill them and get rid of them and degrade them. But there are also microbes that, once they're in the blood, can get into our cells. Now imagine you've got a microbe in a cell and you've got our mitochondria, and basically the mitochondria is off, right? So, and it wants to send that signal as strongly as possible. So it releases some of its DNA, which is a danger signal to the cell. Now our other cells can say, all right, we need to kill that cell. But I just think it's an amazing finding. There's some very interesting studies going on in that, in terms of that, especially specific fatty acids inside that the mitochondria should be using can get stolen by intracellular parasites like toxoplasma. So yeah, we are at it's a continual arms race, us versus them. They're doing stuff to us, we're doing stuff to them. Unfortunately, spoiler alert, they win. We don't make it out alive, besides the 120 years that we, have to date. But it's only a matter of time. Their days are numbered.
So,
it is.
Are you doing any current experiments for longevity in this case with your microbiome? I mean, we talked about the deep sleep aspect. Is there anything up and coming or currently in progress?
Not with the microbiome. I always keep an eye on the blood test stuff because that's the end point. If your microbiome changes, but you don't see any changes in your circulating biomarkers, what does it really mean? But I bet that changes in the microbiome would lead to changes in the circulating biomarkers. So I had my microbiome done a few months ago, but I haven't, as I mentioned, you would have to do it so often to get a true baseline that, I've strayed away from doing it that often and looking at it more once in a while to make sure it's not dramatically changing with age. And in contrast, the circulating biomarkers, I'm still doing that every few months to make sure that my biological age is as young as it can possibly be. I don't know if you saw that post, but I'd say that's it. So Alex Zavaronkov's group at In Silico Medicine, they developed this biological age predictor based on the circulating blood test results, the standard chemistry screening, which when you go to the doctor, it's just 40 blood tests. That simple $35 test. So I'm more hot on that trail. Microbiome is always in my mind. I'm constantly studying it and writing grants about it. But I'm more obsessed about the biological age and optimizing that through circulating biomarkers. So next week, I'll get my blood test done again to see if hopefully I'm still on the right track. But based on that data, I'm about 15 to 20 years younger than my biological age. And what's unfortunate is that, the biological age predictor based on, I think it evokes, the first thought I think most people have is, oh, come on, bullshit, biological age, give me a break. Because the biological age things that have been reported in the past have been based on these ridiculous questionnaires and, how often do you exercise and do you smoke? No, this is based on data from about 200,000 people and using machine learning to develop a predictor set of circulating biomarkers that can predict biological age. So based on that, as I mentioned, I'm 15 to 20 years younger than my actual age. So just trying to keep that as low as possible for as long as I can.
Mm-hmm. Great. So we're just coming up on our time here at the moment, but I mean, yeah, I'm sure there's a treasure trove of Q&A things we could still be going through here. But for any listeners that want to maybe keep following you, see what you're getting up to, the kind of information that you're sharing, or even potentially contact you, are there any particular websites or social media accounts that you'd recommend for people to get follow you?
Sure. I'm Twitter, Facebook. It's at Mike Lustgarten. My academic email is on my papers. If anyone wants to look up my academic papers, I've got my website, michaellustgarten.com. Pretty responsive. So if anybody's got questions or they want to direct me to studies on X, Y, or Z, have at it. Got to keep your eyes open to all the ideas because you never know what may be the right. It's good to listen to all points of view, right? So that's what I try to do, wherever they may come from. So if people want to shoot me ideas and questions, I'm all for it. So.
Perfect. Well, again, thank you so much, Mike, for your time and sharing your personal information and also the information of what the research is saying.
Thanks, Gary. Appreciate it. Have a good one.